Hi everybody, I'm Professor Steve Forsythe, and today I'm going to go through how to change the reference strain in order to do genomic comparisons within the Cronobacter pub MST database. Now I'm going to actually go really up the scale. I'm going to do over 1,000 Cronobacter sakazakii strains in the analysis. And you'll be hopefully interested to see how much information you can easily get by doing this sort of genomic comparison. Now, one of the things to consider why we want to do this is because the Cronobacter genus is now recognized as being composed of 7 species. We've got closely related genera of Cronobacter and Cicibacter, and one has to be very careful on what identification methods one uses. If you want to get more information on this, then please click here with regard to a YouTube video I did on misidentifications, and there's lots of those out there in the public domain and in various refereed publications, unfortunately. So please, if you're unsure about how to correctly, robustly identify Cronobacter, please go to that YouTube video. So the reason for showing this is because in order that people recognize how diverse the Cronobacter genus is, and therefore it's important to choose the most appropriate reference strain. So why change that reference strain? Well, because at the moment, if you just go to the COG MLSD analysis, then you're actually using a reference strain, a default one, which is Cronobacter sakazakii ES15. That's a SD125 clonal complex 100 strain. And if you want to know more about that, then just click down here. It will take you to the published paper that I did a few years ago on this topic. The thing is that it was chosen because it was the first well-annotated Cronobacter sakazakii genome, and things have changed since then. As I say, it's very diverse, and in actual fact, the majority of species are not clinically relevant. So by varying what you're analysing, then it's important, I think, to choose a different reference strain. Now, in order to prepare for this video, for the examples I'm going to show you, what I did, I went to the genome page. I generated an Excel file of the complete database. Now, this is open access. Anybody can do this. So the whole database you can export as an Excel file. I then arranged the Excel file according to species, clonal complex, and sequence type. And there are various videos I've got on YouTube that you can go to if you want more advice on how to do that. This is simply my example. You might well want to be more restrictive and choose a particular country or particular year range or sequence type, etc. This is simply what I did in order to prepare for this video. So in brief, I went to the Cronobacter pub MSD database. The URL is here if you want to click, if you're not sure. I went to the genome collection one. It's a better one, more appropriate to use for this purpose, rather than the isolate collection. You can see how many, well over 1,000 strains we have of whole genome sequence Cronobacter now. So clicked on that. I then scrolled to the bottom of the page, and I then went to this point here. I clicked on where it says database. That then gave me a Excel database, Excel spreadsheet of the complete database, which I then just sorted according to, as I say, species, clonal complex, sequence type. Simply, for my example, you choose what you like to do. Then you go to the genome comparison option of the database, which again is down there at the bottom. I deleted, just clear here, and then copy and paste the selected pubMST ID numbers, not laboratory isolates. Okay, can be confusing, I accept that, but you want those ID numbers of the selected strains. I pasted them in there. On this point of general, I didn't want isolate because you're going to see how complex the diagrams can get. Just for convenience, I just selected like ST serotype. And then this is the the issue here, the important feature is to go to this pull down menu. Is a very convenient thing to do. If you click on that pull down menu, you can see that there's 7 reference strains, and these are like the type strains for the 7 species in the Cronobacter genus, plus there's an 8th one. I've also got an ST4 strain in there, SV291, Cronobacter sakazakii. It was one which was reported to be persistent in the factory, but the thing is that ST4 is also the one that has a very strong association with neonatal meningitis. So it's not the type strain of the species or the genus, but it's a very useful annotated strain of Cronobacter sakazakii, clonal complex 4, which is the neonatal meningitic pathovar. So then you've got the other species here, plus the ST1 strain, the BAA 894. So having clicked on that, just to go step back a moment, what I did for my example, going to show you in particular, I selected the 57 whole genome sequenced Cronobacter tersiensis and universalis as a bit of a variation, and I then use that refsec for Cronobacter universalis. Just a preference, I could have used tersiensis as a reference strain. I used the universalis because I knew that was sequenced using PacBio, so the read lengths are much longer. So just for the example, I chose that one. That took 15 minutes to analyze the 57 genomes. Where you get the output just like this, screen grab of what you get. You then just scroll down and you get these options of various files. The Excel output is you absolutely want that one. There is a graph here. You can click on this figure. We'll see it at the moment. It's great as a quick look, just to make sure that you haven't made any mistakes, which I'm prone to do, I know. So I use this as a quick look, quick error check, but I actually much prefer to then click on this one, distance matrix, next format, click on that, download it, and then I open it in a program called splitstree. And here's a URL for a splitstree. And this is the diagram that you'll get. So this is analyzing 57 genomes. We've got the Cronobacter tersiensis here, and we've got the universalis here. So they're nicely, we would expect those strains of that species to be bunched together and not dispersed amongst the tersiensis. So very easy to do. As I say, it took 15 minutes to analyze 57 genomes. Now that's a nice picture, but it's not really informative. What you want to do is to go to the Excel spreadsheet. I'm just going to show you a little bit of the Excel spreadsheet. Excel spreadsheet, when it did this analysis, that's 3,987 genes you've got listed down here. So you're going to take a while to scroll through that lot, and of course that's your number of rows. Your number of columns is 57 because you've got 57 strains. So this is a huge spreadsheet. It's got all these named genes, almost 4,000 genes for the 2 species, compared one with the other. In this example, then I did the reference strains, I said was the Cronobacter universalis. On the spreadsheet, this column is universalis, this column is universalis. Here's tersiensis, here's tersiensis, and you can see in this region there are genes which are present in the universalis which are largely absent from these particular tersiensis which are in the screen grab. What's of interest is that this region includes the resistance to tetracycline. So it's great because you could then go off to the laboratory, you could take those strains and look for laboratory confirmation of tetracycline resistance or absence in these species or strains of sturciensis, and universalis. Now, you might be saying, but they're not the most important strains clinically, and I agree, but I really think in research it's great to ask the what if, and a fantastic thing about this is that in 15 minutes, you can get a lot of answers to what if questions. I also did the analysis using Cronobacter melanoticus. There's 123 currently whole genome sequence Cronobacter melanoticus in the database. This analysis took 33 minutes. And you can see when it came out on the splits tree that the ST7 are all bunched over here. The reference strain would have been sequence type 7, but the other melanoticus are quite distant on this analysis, which is really interesting in itself. I didn't know this until I pressed the button, took half an hour, then I got this result, and that's something which allow other people to go and investigate. But of course, the title was over 1,000, and yeah, I did a Cronobacter sakazakii. There are currently, as of today, 1,012 Cronobacter sakazakii genomes. This analysis took 15 hours to run, so it took longer than having a cup of tea or a cup of coffee, or even a lunch break. So 15 hours, so ran overnight, and here's the splits tree, which is obviously way too much information for one screen grab, but it's really fascinating that immediately you can see the structure. You can see there are strains which are being clustered together here. There are strains being clustered together over here. And just for the one of illustrating genomic analysis, I'm just going to show you briefly these three areas of the splits tree. If we zoom in over on that left-hand side, we can see that these are sequence type 4. I'm not going to go into any detail of this, but just to show you that you can zoom in according to what information you want. You can find out which strains these are, which country they are that are bunching together. Are they from the same year? Are they from the same country, etc.? Are they the same serotype and stuff like that? Over here, we've got the ones which are more, these are more sequence type 13 over here, and various sequence types over here bunching together. And you can see it's interesting because we've got bifurcating going on. So there's strains which are related. When I say related, I mean in the genomic sense, the DNA sequence sense, they are clustering together. These are a little bit more dispersed, but they're great for one to go and analyze further. And here's a really interesting group of ST1s, complex one, where in actual fact we've got like two branches in parallel, and then we've got this bifurcating going on or division going on. And again, one can zoom in there and have a good look at what's going on and what's driving that diversity. Just zooming in a little bit more, just giving you an example. So the Excel spreadsheet for the 1,000 Cronobacter sakazakii genomes was composed of just over 4,000 genes were analyzed. So there's over 4,000 rows going down this Excel spreadsheet, and there's 1,012 columns going across here from left to right. So it's going to take you more than a couple of days to analyze that sort of output to see what differences, but just skimming over, you immediately see interesting things. So you can just start to skim through. And here's a region, really interesting. This particular strain clearly is lacking a particular genomic region. And when we look at what that genomic region is, we can see primarily it's to do with citrate metabolism. So one could nicely go into the laboratory, get that strain, and then check, do we get in laboratory confirmation of what we predict from the genomic content. So I'm just giving you like a heads up on the tools which are there on the Pub MLST database. They are free to use. This is all open access. There's no subscription. You can do this for yourself or get a PhD student or postdoc to do it for you if you're in that situation. Lots of information freely available, and it's something which I'm noticing researchers are unfortunately not doing, and I think could be really, really profitable. So in conclusion, multiple genome comparison is very, very easy in the PubMST database. You can quickly, really quickly compare strains. One wouldn't normally do 1,000 strains in one run. I just used it as an extreme example just then. The reference genomes are there as a type strain for each species, plus the SP291 as a reference one for ST4. There are others, so you can choose your own reference strain if you want. You just use the refsec file for this, or if you have any problems, just email me. Number of genes being compared now, we're talking about 4,000. In the order of 4,000 genes are being analyzed when you're doing your whole genome comparisons. And it makes use, if you're not studying Cronobacter sakazakii, then if you're looking at Melanaticus, it makes sense to use a reference strain, which is Cronobacter melanaticus, or for Terciensis, use a reference strain for Terciensis. The results, I'm not much of a bioinformatician. I like it because the results come out in an Excel spreadsheet. And this used to be a great way passing on to students and for myself, look through that Excel spreadsheet for genes of interest and then go to the laboratory, look it up. And the other thing is that there's a nice output in a file which you can open up in split screen, you can zoom in and analyze from there. So this isn't the first of my YouTube videos. I've got various other ones out there. There's one which is a two-parter on analysing a neonatal intensive care unit outbreak over 10 years and the variety of methods that we used. There's one specifically on the Cronobacter sakazakii pathovar, CC4. The last one which I did was to do with the strains which are the incidences which have been popularized in America and led to issues over the production of powdered infant formula. So this is an update on the strains of interest. Who am I? Well, I'm a so-called retired professor. Not quite sure what the word retired means these days. Previously advisor for the FAO WHO 5 times on the safe feeding of infants. And you can read all that for yourselves. I've written over 150 peer-reviewed papers, 3 books, all like that, mostly on Cronobacter and infant formula, and many of those papers are involving multi-locus sequence typing. I'm the curator. Here's my email address. Please contact me if you have any issues on how to do this. And finally, I'd just like to thank people who contribute, who send strains in to the database. And the database is hosted by University of Oxford, and the person who oversees it and helps me, give me guidance, is Dr. Keith Jolly. So thank you to Keith and everyone who donates strains information to the database. And there's a couple of references there if you want to read a bit more, and those are active links. So thank you very much for your attention.